homeostasis, donor equilibrium, and these topics we can discuss today. And they may appear in the examination as short notes, or and the oral table may be asked. Apoptosis is also called programmed cell death. By genetic control, most of the cells are dividing and growth takes place, and it is a regulated process, and at the same time there is loss of the cells by genetic programs, spontaneous loss of the cells, and it is called apoptosis. Apo means away, Greek word, and tosis means that is falling down. Tosis, apoptosis. And it is always going on. Some cells are continually being lost, and it is different from necrosis, in which the cells, healthy cells, die during the process of inflammation. But apoptosis is the process like falling of the leaves from the trees. There is no inflammation. It is a genetically programmed cell death. From the development, it is growing on. During the development of the individual, in the nervous system, good numbers of neurons are produced, huge numbers are produced, and during their connections, proper connections, and good number is lost whenever there is the further development and synapse formation. In case of immunity, you are familiar with the behavior of the lymphocytes. In their primary organs, there are huge proliferation of the lymphocytes. In case of thymus, and also Barsa equivalent in the bone marrow, during development there is production of huge numbers of lymphocytes, but there is simultaneous death, so that ultimately the immunocompetent cells are produced, and those clones that are not required and that may cause harm to the individual itself, they are eliminated. Huge amounts of lymphocytes are produced, and those lymphocytes that may harm the individual itself by autoimmunity are lost. Only the lymphocytes that are helpful, and they remain, and they divide in a regulated way. During development, these fingers, they are connected by some tissue waves, and they are lost by the process of apoptosis, and the fingers become separated. There are numerous other examples whenever it is produced. During development of the genital system, the primordial genital ducts, they disappear. In case of whenever there is development of the male, the Mullerian ducts disappear. And in case of females, it is the Wolffian duct that disappear. Initially they are present. And this is the process is going on. In the adult condition, we are having loss of cells. Suppose the enterocytes, the cells that is present in the small intestinal epithelium, and in the small intestine there are villi. finger-like structures, not visible in the naked eye, but under the microscope easily seen. Even low power microscope villi can be seen. And they have their cells, epithelial cells. And the cells that lose contact with the neighbors and the basement membrane, they are susceptible to apoptosis. There is a death of the cells, gradual death, and they were replaced by the cells coming from below. Libercum, crypts of libercum. In your histology text or in your physiology text, and they are written, crypts of libercum. And they are always proliferation. Coming into surface, they disappear. Cyclic blood loss in post-pubertal women is a common feature, menstrual blood loss. And it is also a form of apoptosis. The hormonal support disappear normally. Progesterone action disappears, and then they are slapped out. And from the depth of the cells that are remaining, they proliferate. And in this way, it is going on, apoptosis. Yeah. And during apoptosis, what occurs? Histologically, the cell undergoes some changes. There is condensation of the chromatin, cell shrinkage, condensation of cytoplasm, and then membrane bleb formation. And they are then separated into structures called apoptosomes. And they are cleared by the phagocytes or neighboring cells. There is no inflammation. During inflammation, there are cells, substances from the living cell, they come out, lysosomal enzymes or so, and they causes the process of inflammation. A furuncle is a typical example you are all familiar with. Phora hole dekho na, hate phora hole ki hoy? There is swelling, accumulation of fluid, and there is hotness and redness due to increased vascularity and loss of function. These are the features of inflammation. You will learn in detail, in pathology. And due to some reasons, healthy cells die due to action of numerous toxic substances, including antigen-antibody reaction in the healthy cells. And that is called cell murder or necrosis. Necrosis. But in this case, there is no inflammation and there is loss. And it is replaced by the new cells. It is a process of development. And when the apoptosis is disturbed, then there may be some diseases then that may appear. It may be due to autoimmune disease. There is the immunocytes that are directed against the own cells. They disappear in the process during development. But if this process is not proper, then some cells may remain and it may attain the capacity to act upon the own cells. In this way, autoimmune diseases can take place. In some cases, malignant proliferation or cancer can develop. And in the nervous system, there may be loss of the cells, excess loss of the cells, and some chronic diseases like Parkinsonism, Alzheimer's disease, this type of diseases can develop. Yeah. So apoptosis is a physiological process, and in this way, there is these cells do die by the process within the genetic information. And it is possible with the help of the enzymes that are called caspases. Caspase. cysteine proteins. Cysteine proteins. They break the protein structure up at the C-terminal end of aspartate. Aspartate. N-terminal and C-terminal. And there are amino acids there. There may be this. And it is bound in this case. Caspases. They are present within the cells in inactive form, procaspases. They are activated by this process, during this process of apoptosis. They remain within the cytoplasm in the inactive form, procaspase, and they are activated. And it has a resemblance to that of blood coagulation system. And there are these coagulation factors. They remain within the cells, within the blood plasma in inactive forms. And they are activated by the cascade. Like prothrombin is converted into thrombin. Factor 10 is activated to active factor 10A in this way. 7 to 7A. And there are extrinsic pathway and intrinsic pathway. And in this case also there are two pathways. One is extrinsic pathway, another is the intrinsic or mitochondrial pathway. I can remember very little of them, but I give this little information, and detail you can have in your good text and internet. Extrinsic pathway and mitochondrial intrinsic pathway. Or mitochondrial pathway. In case of extrinsic pathway, there are some chemicals that come in contact with the cells that are undergoing apoptosis initially. And there are receptors called death receptors. DR they are called, DR3, DR4, and in this way. Death receptor. And death receptors have these ligands. It may be tumor necrosis factor or FAS in this way. Suppose a cell that is going into that, it is a receptor. Suppose it is death receptor. And it can unite with substances like tumor necrosis factor or FAS. FAS may be part of a cell, a lymphocyte. It is projection in this way, or it may be in the free form. In some text, you will find that FAS is included as a death receptor, and instead of FAS, they are called FAS ligand. Yeah. But some texts they explain in a more lucid way, in this way. There are some projections of integral proteins from the cells. It may be a T lymphocyte or so, and which can attack another cells. And when the appropriate this structure will combine with it, then there is trimer formation, death receptor, and they are then unite with some adapter proteins called FADD. Suppose in this way, trimer formation, and then it is this TNF, tumor necrosis factor alpha, TNF, and these are the death receptors. and they unite with a protein they called FADD. FADD. Fast associated proteins with death domain, FADD. It may be single or multiple, and it then activate the initiator caspases or procaspases that are in inactive form. In this way, they will activate. And in this case, it is the procaspase 8. It is converted into caspase 8. It is the inactive form, it is the active form. Active. And it then acts upon some substances within the cells, it is the initiator caspase, and then the executor caspase or definitive caspase is formed. They are procaspase 3, 7, they are activated. Caspase 3 and 7, and they have their effects. It is the effector or executioner. And they will have their effect. And in this way, it can inactivate the inhibitor ICD inhibitor of DNase. There are inhibitor of DNase in the cells. Intercellular caspase activated, it is in the inhibitor, and it is then made non-functioning so that the DNase becomes active. Inhibitor of caspase activated DNase, and this inhibitor is suppressed and DNase becomes free, and it causes DNA fragmentation. And procaspase 8, it can act upon the gelsolin that maintains the microfilamental structural proteins, actin filaments or microfilaments. They act upon microfilaments, and so there is this cell structure is lost, and they combine to form this apoptosis. This cell will be then reduced into this part type, and they will be nuclear mass and this type of apoptotic bodies. And there is no element coming from the cell into the outside, and they are called apoptotic bodies, and they are taken up by the phagocytes unceremoniously. There is no reaction in that case. No reaction. এরকম ভাবে খুন করছে যে কিছু হচ্ছে না, কেউ জানতেই পারছে না, পরে তো পোস্টমর্টমে দেখা গেল সে খুন হয়েছে। Genetically programmed, and it is going on. It is going on. Now another pathway is the intrinsic or mitochondrial pathway. There is numerous reasons for DNA damage. It may be radiation, or it may be some other elements, and growth factors, they may be deficient. As for example, in endometrium, the hormonal support is lost, and when there is DNA damage due to any reason, DNA damage, what occurs? Then there is this, ultimately there is the activation of some pro-BAX and BCL2. It is apoptotic and it is anti-apoptotic. and there are mediation of other proteins. There are mediation of other proteins. They may be BIM, BID, BAD like. And ultimately, by their mediation, there is activation of this BCL2 related X protein, and it is pro-apoptotic and it is anti-apoptotic. So ultimately, whenever there is DNA damage, then this type of things takes place. And due to this action of this and absence of action of this, there is cytochrome C is liberated. Cytochrome C is liberated from the mitochondria. Mitochondrial outer membrane is permeable to cytochrome C, and the cytochrome C will come out. Cytochrome C. These are the complex compounds. Chrome means colored, and cytochrome C is active in the oxidative phosphorylation. You have read already. And in this case, and mitochondria is a foreign element into the eukaryotic cell. So it can give some product, but it has some enmity also. In this way, it can remember. It is a double-edged weapon, and it can also take part in the apoptosis or cell death. And cytochrome C then combines with APAF1. Apoptotic protein activator factor 1, and in this way they becomes active and they form an heptamer with, there is this 7 spoke like structures, and in this way they will cause this activation of, in this case there is the pro-caspase 9 to caspase 9. There is the initiator peptide, caspase 9. Or this type of numbers of caspases are present in the cell that has been detected. All of the caspases may not be related to apoptosis. Caspases means cysteine proteases, and there are serine proteases. In blood coagulation system, they are the serine proteases. Yeah. And here is this cysteine proteases. And these are complex pathways. When you go through book, you will find other members, other abbreviations also. So my discussion is a short and give some idea. So with your fresh and better intellect, you can go through books and internet to gain much more better information. Only the basic part I can tell. My knowledge is extremely limited. But ultimately, this is the pathway. Whenever there is DNA damage due to radiation, withdrawal of the growth factors, and in that case, via these intermediate proteins, there is activation of pro-apoptotic BAX and BCL2. Normally there is inhibitor, it's inhibition of this. So there is upper end of this, and there is change in the membrane potential. And due to change in the membrane potential of the mitochondria, from the outer membrane, cytochrome C will come out with the cytosol, and it will unite with a apoptotic protein. And then there is activation of pro-caspase 9 to activation, and there is the initiator peptide. But at the same time, besides this, there are also other inhibitors, or some inhibitors of the inhibitors. In that same, not only this is cytochrome C, also SMAC or diablo will come out. Second, mitochondrial apoptosis, there is a controller also. This type of protein also liberated from the mitochondria, and it will causes inhibition, some inhibition of some inhibitor. inhibitor of apoptosis proteins. So ultimately, there is formation of this structure, compound structure, and there are a wheel-like structure, 7 spokes in this way, multi-bar formation. And they causes apoptosis. They pro-caspase to caspase 9, and from caspase 9 it will activate, there is the pro 3 and 6 to 3 and 6. That is the executioner peptides. The same caspase 3, and there is this, in this case this. So there is a link between them, and there is a common pathway, and this is the common pathway that apoptosis occurs. Apoptosis occurs. Yeah. Common pathway. That is the DNase is liberated. By inhibiting the IC inhibitor of DNase inhibitor, and that is inhibited, and DNase will be active, and it will cause DNA fragmentation. At the same time, the structural component of the cells, microfilaments or actin filaments, their structure is also destroyed by acting upon this gel solid, and then there is this type of reaction occurs. The cells will ultimately, initially that becomes condensed, and there is mitochondrial condensation, there is a nuclear condensation of the nucleus, changing the shapes, and the cytoplasmic condensation, and membrane bleb formation. Ultimately, it will break down into these structures called apoptosomes, and they are taken by the phagocytes. And in these both these pathways, it will take place. So this is something regarding apoptosis. So if there is failure in the apoptosis, there may be autoimmune disease. Immunocytes acting upon the own cells may be active. Autoimmune disease. There may be neurodegenerative diseases. There is early destruction of some cells, unwanted early destruction of some cells of the neurons, and chronic disease can occur. Or in some cases, there is the increased growth of the cells, abnormal growth of the cells, leading to cancer or malignant or tumor formation. And at the same time, in this case, there is a protein called P53, and it has a role in some way. It can cause inhibition of the mitosis whenever there is some destruction. There is some disturbance in this. P53, role of P53 protein, pro-apoptotic protein of P53. And absence of P53 protein is noted in a good number of malignant tumors. So it is a brief and incomplete deliberation from me, but if you will have listened this, then you have some idea. There is two pathway similar to that of the blood coagulation pathway. One is the intrinsic pathway, other is the extrinsic pathway. In this way, it is the mitochondrial pathway, and it is the intrinsic, and it is the extrinsic pathway. And in the pathology text, you may find there is intermediate, it is a process. One side apoptosis, one side necrosis, and in some cases there are intermingling. It is some elements of apoptosis and some element of necrosis. In pathology text you can find. I am not going into the detail because my knowledge is limited. I will confine into only the basic physiology texts. Clear? Now homeostasis. And what is homeostasis? You all know about homeostasis. That when an individual or an animal or anything living thing, it has a steady state. Yeah. Suppose we are on living creatures, and our body fluid volume is more or less appropriate to body size, and its hydrogen ion concentration, sodium concentration. What is the pH of human plasma? What is the pH of human plasma? And these are all you have learned in the school days. School days. In the higher secondary text, what we say, all are written in higher secondary texts. And when I go through higher secondary texts, I think, what is the utility of keeping this subject alive? They have, the meritorious students, they have learned this in the school days. And you all know that it is, in case of arterial plasma, it is 7.40. 7.40. It is an accepted value. Yeah. And when it becomes lower than this, then you call it a acidosis. And when it is more than this, you can call it alkalosis. And there is a range, not a good range, narrow range within which we can survive. Clear? From 7.0, roughly for 7.0 to 8.0, our life is possible. But beyond that, very difficult to survive. And nature has every reactions to change so that it bring back to normal. Suppose if there is acidosis due to any reason, acidosis due to any reason, then there is metabolic acidosis, you may call, diabetic acidosis, and there will be increase in ventilation. Increase in ventilation. Yeah, ventilation. Or during alkalosis, if you drink good amount of sodium bicarbonate, and then the respiration will be to some extent suppressed, so that the carbon dioxide can accumulate to some extent, so that the set point is regained. So it is the, now from this you can have the idea that our body systems are formed of cells, cells from tissue, tissues from organ, organs from organ system, and all the organ system come together into a single structure, that is called the human body or animal body, whatever it means. So every cell must have its environment optimum for its work, and every cell is surrounded by which fluid? Every cell is surrounded by which fluid? Every cell is surrounded by extracellular fluid, better interstitial component of the extracellular fluid. So it is the nature's duty to bring its concentration more or less normal, so that it can have its function. In the interstitial fluid, sodium concentration, what is the sodium concentration in the extracellular fluid? তোমাদের কাউর এতদিন তো হয়ে গেল, তোমাদের কিছু অনেক পড়া হয়ে গেছে অনেকটা, হ্যাঁ? 142, what is the unit? মিলিঅসমোল। সেটা ছিল না, তোমার এটা মনের মধ্যে রাখতে হবে, সেগুলো বুঝতে পেরেছো? সব ভুলে গেলে চলবে না। এটাও তো বলেছে, এটা হচ্ছে 142 মিলিমোল পার লিটার or milli equivalent per liter, electrical equivalent, because its charge is 1, valence is 1. Or here there is the milliosmol per liter. It is also to some extent correct. So he has gone through books, and I all request you, very easy. Physiology is a very easy subject. Physiology is a very easy subject, and those who service in physiology, they are considered a lower category of people in the hospitals. If you in clinical side, if you mention our names, they will be irritated. Why you are talking of those inferior quality of people? So we are considered as lower people. In spite of that, we are servicing. It is our livelihood. So you have to go through books and have your proper knowledge. Now these homeostatic mechanisms, there are some mechanisms by which this stable state is maintained, and it is disturbed in many ways, and there is every attempt of the system to bring it back to the normal set point. And normally this is by there is a set point, and it is then when it is altered, there are some sensor that is activated, and this sensor will causes change in the physiological processes so that it can be brought into the normal value. Normal value. Clear? Set point. Body temperature. Body temperature means that is the core temperature. Surface temperature can vary, but the core temperature remains normal and is a relatively fixed value. 36.8 degrees Celsius, that is close to 37 degrees Celsius. And whenever it changes due to some reason, the nature tries to bring it back to normal. Suppose during hot days, what occurs? During hot days, what occurs? Then there is sweating. When the external temperature is close to the core temperature, even more than that, then what occurs? And in this way, the body will gain this heat. The process of heat transformation, heat transfer from one body to another, it is a part of physics that you have read. There is the conductance, convection and conduction and radiation. Yeah. Suppose when the environment temperature is 40 degrees centigrade, then in every way heat will be come into the body. And what is the way that body can rid of? It is only evaporation of water. And by the different receptors present at different site, they send information in the hypothalamus, and ultimately there is sweating and peripheral vasodilatation. During sweating, there is called latent heat that is lost. Latent heat. So this, whenever there is going in rise temperature, there is excess sweating, and it is the process by means of which our life is possible. And if there is this process of sweating is disturbed due to some disease or due to some environmental factors, then there is hyperpyrexia. And due to this hyperpyrexia, there is damage of the nervous system, and that can be death in that case. তীব্র মারা যায় না, প্রচুর লোক আমাদের দেশে মারা যায়, বুঝতে পেরেছো? মারা যায় ডিউরিং সামার ডেজ। এর জন্য কৃষকরা কি করে, জায়গায় জায়গায় গাছ রেখে দেয়, তার ছায়াতে গিয়ে যেন বিশ্রাম করতে পারে, বুঝলে তো? Common people has little value of their life, so whenever they diseased, mostly they get no treatment or very inferior quality of treatment, and they die. পূরণীয় ক্ষতি হয়, ওদের অপূরণীয় ক্ষতি হয় না। সাধারণ লোকের মূল্য খুবই সব দেশেই খুব কম, but they want to survive, and for this reason they plant trees, larger trees that have their shadows, and they can take rest there, and drinking water must be available at this, and some salt may be available. And if there is these things occurs, heat exhaustion or heat hyperphagia, then he should be brought to the shadow and given plenty of drinks, and also so that, and also the movement of the air by any means, so that evaporation can take place rapidly and the individual may survive. Salt drinks and also the drinks, simple drinks, water can lead to survival. On the other hand, during the cold days, and you see that the shivering takes place, there is vasoconstriction, and there is movement of the muscles, involuntary movement, and in some cases semi-voluntary movements can increase. There is every attempt to increase or that is the body temperature, so that the loss is avoided. We put on extra clothes so that heat loss can be avoided. In this way life is possible. And in case of animals, you see, there will be increase in the thickness of the fur. The hairs stay on their ends and there will be thickness of the fur. By means of smooth muscle, erector pili muscles, there is thickness of the ears. There is the ear layer will increase. বিড়ালের তো অত সুযোগ নেই যে একটা পোশাক পাল্টাবে, যদি তার কোনো সেরকমই থাকে। তার যদি কোনো হিউম্যান ভলান্টিয়ার না থাকে তাহলে সে কি করবে? তার তো বেঁচে থাকতে হবে। অ্যান্ড নেচার ইজ ইন্টেলিজেন্ট এনাফ, তাকে সেটা দিয়ে দিয়েছে। দেখবে বিড়াল কেমন শীতকালে গোল হয়ে বসে থাকে, কারণ হেয়ারগুলো সেটা এন্ড হয়ে যাচ্ছে। এবং the thickness of the air is also directed by the temperature, external temperature. So these are the examples. That is the how it is made stable, and it is the negative feedback. Negative feedback. That is, a set point is disturbed and it is brought to this near the set point, so that survival is possible. Negative feedback. There are numerous examples of negative feedback, and major part of the physiology text can explain, can try to do this. Suppose if there is hypoglycemia, and it will be detected by some organs. Besides nervous system, endocrine organs can take place. Hypoglycemia leads to increased secretion of glucagon and catecholamines, and they will try to bring back this glucose level to normal. Yeah. Calcium concentration can go down due to some reasons, and in that condition, there is the lowering of the calcium is then it is parathyroid glands will come into action. It will liberate parathormone and 84 amino acid peptide, and that will act upon the bones and to some extent in the intestine, so that the calcium can come from the stores and from the food source into the body fluid, so that it is maintained. If there is much fall in the calcium ion concentration, then there will be involuntary contraction of the muscles. There may be respiratory arrest and death. So parathormone is an essential hormone in that sense. Yeah. Calcium ion concentration when go down, the condition is called tetany. T-A-T-A-N-Y. That is involuntary contraction of the excitability muscle, involuntary contraction of the muscles. And that can involve also laryngeal muscles. And if the laryngeal muscles can contract, and then there is every chance of asphyxia. That is, ventilation will not be there. And in the asphyxia, within a few minutes, the individual can die. So the killers can kill their enemies in this way, by the process of strangulation also. Within a few minutes, they become successful. Similar thing occurs due to this disease. And so parathormone will help in this way. So parathermone is an essential hormone. Many hormones in the long term, if they are deficient, they may have some deleterious effect that may lead to death, but it is a chronic process. But acutely, it is the parathormone. It is the parathormone that is. So parathormone is an essential. These are the examples of negative feedback. If there is blood pressure rises, then there are baroreceptors. Baroreceptors will come into action. They are present in the large arteries, carotid, sinus, aortic arch also. And baroreceptors work in this way. They will suppress the sympathetic stimulation and activate the parasympathetic system, so that ultimately there are the fall of blood pressure. If the high blood pressure remains for a considerable length of time, there may be some rupture of the cerebral vessels. Cerebral hemorrhage can take place, and in this way the individual may die. So nature is protecting hand. Whenever there is fall in blood pressure, same baroreceptors will act in the opposite way, so that the blood pressure is coming to the, that is the normal level, close to the normal level. So this life is possible. If it is not possible, the condition goes into the shock stage and the individual may die. So there are major part of this physiology is directed to this control. It is the negative feedback. But in some cases, there is a positive feedback. Positive feedback. Positive feedback. Whenever there is a change, this change does not return to the normal level. The change will produces more change in the same direction, same effect, and ultimately the system becomes destabilized. And it may be detrimental in some cases, and it may be beneficial in some cases. Clear? It is a positive feedback. যে চেঞ্জটা হয়েছিল সেটার অন্য এবং সেটার বেড়েই যাচ্ছে। একবার চেঞ্জ যখন হতে শুরু করলো, সে তখন আউট অফ কন্ট্রোল হয়ে যাচ্ছে, কমপ্লিটলি ডিস্টেবিলাইজড। অ্যান্ড ইট মে বি বেনিফিশিয়াল ইন সাম কেসেস, অ্যান্ড ইট মে বি lethal in other cases. Suppose our heart muscles are getting blood via the coronary arteries. Coronary arteries at the very early branch of arch of the aorta, first branch, right and left coronary artery. Now if there is some, initially there is some due to atherosclerosis or something else, then there is less blood supply. The heart muscle may suffer, and if there is ischemic damage, then its power of contraction will be reduced, and whenever power of contraction, then blood pressure will fall. Less amount of blood is going through the coronary arteries, the muscles will suffer more, and in this way a vicious cycle will develop. Ultimately the cardiac output will be so low that this life is not possible. It is the reason for the death of the myocardial ischemia. মানে হার্ট অ্যাটাক হলে মানুষ মারা গেছে, কেন মারা গেছে? এই কারণেই হচ্ছে। বুঝতে পেরেছো? পজিটিভ ফিডব্যাক in this way can go on, and it is lethal, but in some cases it is beneficial also or physiological. Suppose whenever there is action potential generation, action potential is a physiological mechanism by which life is possible. কেন অ্যাকশন পটেনশিয়ালটা এত প্রয়োজন? কেন প্রয়োজনটা হচ্ছে এই কারণেই যে সেই ক্ষেত্রে এই যে রেসপিরেটরি মাসেল, দে আর দি ভলান্টারি মাসেলস, and they are regulated by the respiratory centers, and the nerve impulse will come, always coming, and for this reason we can respire and keep alive. So if there is no action potential in that case, then life is not possible. So action potential is important. Now what occurs in the resting membrane potential? That is the, in case of muscle cells, inside and outside there is potential difference. বিভব প্রভেদ আছে, বুঝতে পেরেছো? Normally cardiac muscle, you find in the books, it is a minus 90 millivolt. Minus means inside in comparison to outside. Now, if there is some change in the membrane potential, then some voltage gated sodium channel will open and it will be partially depolarized. And whenever this partially depolarized, more sodium channel will open and there will be more depolarization. And in this way, it is cumulative effect. So ultimately point is reached, that is the firing level. Whenever there is the sudden opening of the voltage gated sodium channels and they will open. And there is action potential generation. And they will spread from one cell to another. One cell to another in case of cardiac muscle. And in skeletal muscle, by means of these nerves, every muscle cell, skeletal muscle cell, individual innervation. And whenever there is action potential generation, then this nerve impulse will come and what will occur? There will be liberation of the neurotransmitters. And neurotransmitters will cause depolarization, ultimately action potential degeneration in the skeletal muscle cells. And in response to this action potential, calcium is available and the muscle can contract. So it is an example. Suppose in case of ovulation, in the middle of the menstrual cycle, on the 14th day or so, then there is post-pubertal woman and ovum is liberated with the help of LH surge, luteinizing hormone from the pituitary, luteinizing hormone. Normally, in normal conditions, that is the estrogen that is liberated from this effect of luteinizing hormone. It causes inhibition of this luteinizing hormone. But for certain reason, not better explained in any text. Now, then there is positive feedback at the near the middle, close to the middle, and there is liberation of estrogen, and estrogen will positive feedback. More luteinizing hormone will be liberated, and more estrogen will be available, and it will again its positive effect. In this way, there is the LH surge, and in response to this, there is a liberation of the ovum, liberation of the ovulation, and it is a positive feedback mechanism. Positive feedback mechanism. Childbirth has the same type of examples. When the fetus is going through, and there is the role of a hormone that is called oxytocin. Oxytocin. And when it is going through the passage, then neural impulses will come, and it will liberate more oxytocin from the posterior pituitary. And this positive feedback process goes on till the delivery of the baby. Similarly, in the process of digestion, that is the breakdown products of protein and fat, causes liberation of this hormone that is called cholecystokinin, pancreogymin, or simply cholecystokinin CCK. So what occurs? Within the stomach, there is some digestion of the protein and minimal digestion of the lipids, and they will come into the part in the small intestine, and in response to this, there is liberation of cholecystokinin. In response to cholecystokinin, more enzymes will be liberated from the pancreas, and the pancreatic enzymes, they contain lipase and also proteolytic enzymes. In response to this, there is more digestion of the protein and lipid, and their breakdown products will increase. There is more liberation of cholecystokinin and more availability of this enzymes, pancreatic enzymes, and this process continues. It is an example of positive feedback, and in this way it will go. And positive feedback is also seen in the coagulation system also. Initially, whatever with the extrinsic or intrinsic pathway, initially there is some formation of the thrombin, and thrombin that is a positive feedback effect upon some coagulation factors. Their active form will increase, and in this way, ultimately, it is enhanced. The process is enhanced. It is also an example of positive feedback, and they are beneficial. In case of myocardial infarction, in that case, it is but in this case it is beneficial. So these are the mechanisms, negative feedback and positive feedback, and these processes are operating, and they are always operating, and in this way it is maintained. In all the systems you will find, all the systems. Yeah. And there is a term that is called gain in this case. In negative feedback system, there is a term called gain, and it is a numerical, it is a measurement by means of which you can understand how it is operating. Gain. In the text that I requested you, and they give an example. Suppose an individual has a normal value. Normal value of 120 millimeter of mercury, blood pressure. If the individual is then infused, then the pressure will go up to 150, and it will have the feedback effect, negative feedback effect. Suppose it is 130 is the resultant effect. Then there is the gain. It is this error, and it is the correction. So correction is 150 minus 130, and error is 130 minus 120. So it is 20 by 10, or 2:1 is the ratio. It is called gain. Yeah. Or it may be the total requirement divided by the correction, divided by the error minus 1. In the same result you will get. In this way it is gain, the concept is gain. In case of positive system, it is, that is the secondary response divided by the primary response. Suppose in this case, if due to some cardiac disease, it initially goes to suppose 110, but it is not reverting to this normal, it is on the same change. Suppose it is then 100.05. It is the secondary change, it is 5. And it is the primary change, it is 10. So in that case 5 by 10 is the ratio, or 1 by 2. In this case, positive feedback. And if it is more than 1, then there will be vicious effect. Or if it is less than 1, it may attain a value in which life is possible, or self-limiting. Yeah. And you will read yourself. So nodal equilibrium, and this part we will discuss the next day. Membrane potential. And you have to go through books, and when you read, you will say these are extremely easy things. In spite of that, we are doing this, taking this, as our directed by the university.